Nine million people living in the coastal zone of Southern California create a number of problems. In an urban society, we require a great deal of energy, electrical energy. The production of energy has costs. The benefits of energy are enormous, but obviously there is a cost to the environment. I think the important thing to remember here is that in environmental effects, none of them are simple. This particular study involves an attempt to produce the best possible technology in an electrical generating facility along the coastline. Electrical generating facilities have two sorts of problems when we speak of environmental problems. First, they take water from the ocean, move it into the generating plant as a source of heat exchange, a cooling source. And within this water mass are organisms, marine organisms. Many of these organisms will be killed in this water movement. This is titled entrainment. The second effect is the effect of heating the cool waters, producing a heating effect on the surface of our local marine environment. We're going to run a series of about 15 to 20 trawls at different times of day at different depths to see if fishes are moving between different depths at different hours of the day. We're particularly interested now in the populations around the intakes at El Segundo and King Harbor, both generating plant intakes in Santa Monica Bay. And we're interested in the schooling populations that occasionally move into the intakes, become entrained by these intakes. The structure of an intake and discharge system in an electrical generating plant looks approximately like this. Intake is located about 18 to 30 feet below the surface of the water, takes cool water into the plant through approximately a mile-long pipe, enters a screen well where the water is filtered through a traveling screen system. This takes out larger organisms from the water. The screened water then passes into the heat exchanger in the generator, where it exchanges heat with the coolant water in the generator itself, and then is discharged through a second pipe, usually discharging at the bottom somewhere offshore. The discharged water is much lighter, it's warmer, less dense, therefore it comes directly to the surface and usually flows as a rather thin surface layer immediately above the discharge. Often a heated effluent in the area of a harbor seems to serve as a nursery ground for young fishes which are preferring a warm water condition. Originally, intake pipes, which are approximately 14 feet in diameter, were simply vertical pipes sucking water directly from the water column. This kind of pipe entrained everything that swam over the pipe. The deflection of water directly in a vertical pattern seemed to entrain everything. Subsequently, designs were developed which transferred the movement of water from vertical to lateral or horizontal. The second discharge system is typical of all Edison plants, most Edison plants in the Pacific Coast, which has an intake cap, which then changes the direction of the flow to a lateral flow. About 90% of the organisms that were entrained in the original intake are not taken in this second installment. The new San Onofre plant, the nuclear plant, will have a third model which has expanded the diameter of the cap so that the velocity of water entering the cap is much lower, even though the total volume of flow is much greater. And by decreasing the velocity of the water, they will decrease the likelihood that organisms will be entrapped in this moving water mass. The purpose is to find a way of eliminating this problem of entraining fish in the intakes. So we're trying to find out at this point what these populations are doing over an 18-hour period or 24-hour period, see if there are certain times of day where entrainment is more likely than others. We've been using a 25-foot balloon trawl. The first trawl here was just to the north of King Harbor, and in 12 fathoms of water. Now we pull the net for 10 minutes, bring the net to the surface, and then all the specimens are examined. We are counting the number of organisms, identifying all the species that were taken in each area.
That crab, Bill.
It's cancer and no claws. Somebody's already had him for dinner.
Really?
Cancer beats me.
Cancer productus, probably.
We're trying to determine the size distributions on these fish. So at this point, we're just looking at everything that we've picked up, and we're counting it, and we're measuring everything, so we have a good ratio of lengths. On many of these species, we've already worked out an age distribution according to length, so if we have just the lengths, we can talk about the age distribution in the population. We're going to take samples home and check the sex of each individual. We'll be returning most of these for the lab to study fecundity, the number of young, how ripe they are, whether they're ripe females or immatures. We'll see if they're spawning.
200.
This project that we're working on here is to just remove the eggs and the larvae from the plankton samples which we take at King Harbor.
As you can see, there are eggs of different sizes and different characteristics, and this is how they appear under the microscope. Some of these can be used for identification, numbers of oil globules, and the characteristics of the membrane are sometimes commonly distinguishing. These are just a few millimeters in length right now, but the fish that we all see in the ocean that we catch as sport fish that we consume start their life cycles as organisms that are approximately this size. The idea behind gathering the information is twofold. First of all, to get an idea as to the species of fish, if you can get that precise, that are in the area, and then also to see how these factors change with time, and to determine whether the populations are being affected by the thermal output from the steam generating plant.
Occidental is emphasizing a combination of academic instruction in the area of marine biology and work study. We not only train people with theoretical aspects of marine biology, but we put them in the field so that they're actively trained in the subject. Our students leave the college with the ability to perform research in a field environment.
My impressions usually in the morning are somewhat dulled because of my own mental state. Just sort of stumble on board and go find someplace comfortable to sit down and relax until the actual activities begin. It's one of the bad things about scientific research is that a lot of it, naturally, you have to adjust your activities with that of the organisms that you're looking at, and unfortunately that means getting up early in the morning and sometimes working till late at night.
A lot of students don't have a chance to go out and to actually see what a fish is or what invertebrates are. They just read it in books, and this way they have just physical contact with it and getting field experience and being able to apply that later on.
I think that Southern California Edison is getting something unique. I think in the long run they're getting better data and better results and more results for the time that's put in. And no, I think it's a great deal. I think it really works.
On board the Vantuna, there is no segregation as to male or female. Everyone has to pull their own weight. I'll be pulling the nets just along with everyone else, carrying the bottles, washing buckets, scrubbing the deck. When you're on board, you're one of the guys. You're a man, essentially. I think in the entire oceanography program that we have here, I enjoy working with the people and having the opportunity to show them exactly what the marine biological environment is.
Very often, students for classes or independent studies or something, they need fish samples that they can work on, live samples that they need here. So we have a cold room set up that's refrigerated at a constant temperature. It's about 50 or 55 degrees to keep the water temperature constant. And we keep live fish back there in tanks, and we can either just keep them in the tanks and then use them for studies somewhere else, or we can just set up a study in the back room.
We're sampling intakes with nets of a variety of sorts, with acoustic sounding and from diver surveys and photographic surveys around the intakes both day and night. We have 2 sets of gill nets set adjacent to the intake. Fishes that swim into contact with the gill net usually are captured by the net. These nets are tended every 2 hours by Occidental students, 24 hours a day for an 8-day period, and we therefore can record the number of fish that come close to the intake every 2 hours throughout this period. The Seeker is running with some people from the University of Washington Applied Physics Laboratories, which have been studying methods of determining or counting fish stock using hydroacoustic data, kind of a sonar tracking system. And they have their machinery now, and they're running in front of us with this hydroacoustic device. They'll monitor fish, but all they can see is fish shadows. They're getting the echoes back from the fish. They don't know what they are. Fishes of different sizes and different species should cast different echoes. So we're following along behind them, and when they tell us to, we will drop the trawl and trawl over an area where they feel they're monitoring fish. Okay, then we'll see what kind of fish we got and how the number of fish that we catch compares with their acoustic record. The Trojan is a Lampara netter, and a Lampara net is a neat way of surrounding a school identified by the acoustic boat, and therefore you can identify the fish seen in the acoustic record and check its accuracy. The Lampara net is a deep net that drags the bottom. In an area where the bottom is not strictly sandy, a diver must tend the bottom of the net to keep the lead lines from tangling on obstructions. The point is that in many areas, visual counts are impossible because the water is just plain too dirty. This is especially true at San Onofre, where it's almost impossible to do any visual monitoring. And so we would hope that we can develop a system through the Applied Physics Laboratory that could tell us the sorts of fish that we're dealing with and their numbers in and around the intake, so we could monitor almost constantly around intakes with some sort of hydroacoustic device. The other thing is that we really don't know very much about the movements of fish populations on the coast here, and this is the ideal device to use. It's so much quicker. It's all recorded on tape, goes into the computer at the University of Washington, and they come out with a printed pattern of distribution. The only problem is they don't know what they're looking at. We're dropping right now, so they must have given us the signal that they're monitoring fish. Go ahead and drop the board. This is a combined study involving a number of groups besides Occidental College and Edison personnel, the University of Washington Applied Physics Lab, and Dames and Moore Consulting Group have been intimately involved in this project.
Okay.
What we see primarily is that fishes around warm water discharges are very abundant and very diverse, and we see very little, if any, detrimental effect from hot water discharge. In an examination of the fishes that are caught in intakes, it turns out that about 6 species make up the majority of the catch. We're trying to find out why these fishes are entering the intakes, what factors are influencing this movement from the water column so that we can avoid it in the future.
This is, I think, one of the really unique aspects of the program at Occidental, in that this is an active program. We have lots of students involved in it, and it gives them an opportunity to get into the field, become actively involved in the collection of specimens, processing of specimens, collection of data, interpretation of data, and writing of these reports. There's really no substitute for actually doing it. You can sit in the classroom and talk about it as long as you want, but it's, until you really get out in the field, like we were doing today on the boat, and really get your hands into some of these fish and handle them and some of the practical experiences. And I think this is probably the big benefit for most of the students.